Okay, welcome my dear friends. This is Dr.PADALA Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal. Still, we are going to continue our CPGET 2023 free online coaching, especially for the young aspirants, those who are going to attempt your PG entrance examination for the Telangana state different universities, including state first women university, that is a Koti Women's University, and other 6 universities will going to conduct the state level common PG entrance examination 2023. In this regard, Botany department from different colleges are going to associate to conduct this online coaching, free coaching, especially Government Degree College for Women, Jagityal, and SRR Government Arts and Science College, Karimnagar, Government Degree College for Women, Karimnagar, and RBVRR Women's College, Autonomous. These are the colleges going to associate to conduct this free online coaching, along with the banner of Telangana State Degree College Association, TGCGTA, Telangana, General Secretary, respected Dr. Kadaru Surendra Reddy Garu will going to planning before starting this valuable session, organize this wonderful program for the young aspirants. In this regard, the state various colleges experts like today's our eminent resource person, they are acted as a resource person for delivering their lectures, their valuable, giving their valuable time for us for better future of the young aspirants. We are very thankful to all the resource persons of various colleges, resource persons, very huge experienced personalities, and very humanitarian services personalities are involved in this wonderful program, my dear friends. So today's our eminent resource person, very kind-hearted and very friendly nature, Dr.A.Madhuri Madam Garu, Assistant Professor, Department of Microbiology, Government Degree College, Autonomous, Government Degree College, Autonomous Women's College, Begumpet. Madam, please welcome and share your screen and start your session, madam. Please welcome.
Hello, sir.
Yeah, you are audible.
Some emergency. I need some 5 minutes break.
Okay, definitely. Every day we are sending one assignment or quiz or the practice test. Better you have to utilize that attempt that quiz or assignment, even though the practice test. That is useful for your CPGET entrance examination whenever you are attempting the examination, because of a specified questions and how the questions are framing, how to answer the correct answer. So these are the main important framing you are observed, though whenever you are attempting this quiz. So better you have to utilize that quizzes and practice tests and assignments. By today evening or tomorrow, definitely we will send previous question papers and analysis of the previous question paper. YouTube links also will going to send in the WhatsApp group. Better utilize this opportunity, my dear friends, because of we are giving in the free online mode, my dear friends. So sparing their valuable time for us. A very experienced and very senior most personalities are involving as a resource person in this wonderful program. So my dear friends, we hope you definitely utilize this program, and also I hope this program is meant to reach your goal, I hope. And also some of the participants already sent in my WhatsApp group their PG entrance registration form. And also, whenever you are going to download your PG entrance hall ticket, please send my personal WhatsApp. And also some of the participants, they are preparing for the jail, DL, and other PPT and Gurukula jail. So, my dear friends, better you have to send your online application form, that is the registration form, because of that is useful for my documentation. No need to send any other person, my dear friends. That is purely confidential. I don't send any other persons. So that is purely confidential because of this is a pre-online mode. So we are all giving such a wonderful program. So that's why you sent your online application form or even the hall ticket. Don't forget, my dear friends. Okay, madam. And my dear friends, 20 video links are there. 20 video links. That is purely CPGET previous papers analysis video links, my dear friends. So by today itself, every day, one video link will send in our WhatsApp group. Better you have to utilize how question is coming, how question has came, how to which one is the right answer, and another 3 options also I clearly explained. So better that is useful. I hope that is very useful for you. Okay, madam, are you here?
Shall we start?
Yes, madam. Yes, please welcome and start your session, madam. Please welcome.
Good evening, friends. Today's topic is Archeabacteria. Arche means old. The meaning of Archeabacteria means old. Okay. So earlier, as we know, there were many types of classifications. There used to be kingdoms, and at the last they have divided these. First, they used to keep what do you say, eubacteria and archaebacteria into the same group, that is monera, and then they have separated about eubacteria and archaebacteria. U means true, okay, true bacteria. Archaebacteria means, archae means old, okay, or ancient bacteria, right? Now we have 3 domains, that is bacteria, archaebacteria, and eukarya, right? So this is basics about classification. Now basically what you have to remember about archaebacteria is, archae means ancient bacteria. These are, as we are saying, they are ancient, or they are first living forms which are found on the earth, right? So they will be more primitive. Primitive means not well developed, and they are having unusual cell walls, membrane lipids, ribosomes, and RNA sequences. Like their structure is not similar to normal bacteria, right? And their specialty is they can survive in extreme conditions. Okay, archaebacteria means they can survive in extreme conditions. It can be extremely high temperature, or extreme salty, or extreme acidic conditions, or anaerobic conditions, and they have the ability, some of them can also produce methane. Okay, so we call them as methanogens, right? These are some of the important points about archaebacteria. This is, they're saying they have discovered this archaebacteria from Yellowstone National Park's hot springs. Hot springs means the temperature will be very high, boiling temperature. Okay, from there they have isolated this archaebacteria. We feel like there can't be any living forms, but in such conditions, these archaebacteria have been isolated. And as we have discussed, they are present in extreme environments. It can be hot springs or salty lakes, and even normal environments also they are present. And they are unicellular. They are unicellular, means single cell is present. And one more important feature is peptidoglycan is not there. If you see the structure of normal bacterial cell wall, peptidoglycan will be present. But in archaebacteria, peptidoglycan is absent. And they do have flagella for their locomotion. And then they don't need oxygen to survive. Means they are called as anaerobes. right? Some of them are, mostly they are anaerobes. They don't need oxygen. They can produce ATP from sunlight, means they are doing photosynthesis. They can survive enormous temperature extremes, and they can survive under rocks, in ocean floors, in these unusual conditions. They can be present, and they can tolerate huge pressure differences. If you see the ocean bed, okay, the organisms which are present at the ocean bed, they have to bear a lot of pressure on them. Okay, they are called as barophiles. Next, size, they are very less, 1 micron long, and we need electron microscopes to see these Archeabacteria. And coming to the shape, they can be rod shape or round like cocci, and even spirillum. Normal bacterial shapes are found in this Archaea. And coming to locomotion, they have flagella for the movement. And cell features, they have cytoplasm, DNA, and the plasmids, but the cell wall is semi-rigid. Okay, in normal bacteria, peptidoglycan will be there, so cell wall will be rigid, not semi-rigid in normal bacteria. But in Archeabacteria, cell wall is semi-rigid. Okay, and why the semi-rigid is, it is to maintain its shape and chemical balance. For maintaining the chemical balance, they need this semi-rigid cell wall. And in membranes, they are having phospholipids. If you see the structure, this is phosphate group, and this is glycerol, and this is lipid. Okay, so these molecules, they are having glycerol ether lipids, and this kind of bond, ether bond, is giving them possibility, or this bond is giving them flexibility to survive in extreme environments. Understood? So which structure is responsible for their survival in extreme conditions? The ether bond present in their membranes. Okay, glycerol ether lipids. Next, this Archeabacteria is divided into 5 phylums: Crinarchaeota, Euryarchaeota, Corarchaeota, Tomarchaeota, and Non-Archaeota. And no need to get into details. Just I'll be discussing only brief about these different phylums. Okay, Crinarchaeota, they are irregular in shape, means their shape is not rod, cocci. They can exhibit different shapes. Single organism, they can change their shapes. Okay, they usually are thermophilic and hyperthermophilic. Thermophilic means to some extent they are at a higher temperature. Hyper means more higher temperature. Sulfur metabolizing archaea, they can utilize sulfur for their metabolism. And they are gram negative, and morphology is different. And in this phylum Crinarchaeota, one of the famous organism is Sulfolobus sulfatericus. It can grow at 80 degrees and pH 2 to 4. Next, Euryarchaeota. This is having 7 classes. We can see Methanococcus, Methanobacteria, all these are methanogens, like they are producing methane. Okay, they are methanogens, they are extreme halophiles, means high salt conditions they can survive, and they can utilize sulfur for their growth. And extreme thermophiles. Next is halophiles. Halo means, salt lake. Okay, in salt lake at Utah, these microorganisms were found. Examples is Halobacteria. Next, methanogens, they can produce. Gen means production, methane production. Okay, they are strictly anaerobic, and they occur at the bottom of lakes, swamps, where oxygen is not present, and they are also present in the gut of herbivores like cows. Okay, they in the intestines of the animals, and they also grow on dead and decaying organic matter. And these microorganisms, they are utilized for biogas production in bioreactors. Examples, this given in the bottom. Next is thermophilic and sulfur reducing archaea. Thermophilic. Philic means loving. Thermo means high temperature. Okay, the organisms which love high temperature are called as thermophiles. They require hot water for their survival, and these are of two types: thermoacidophiles and hyperthermophiles. Thermoacidophiles, thermo means heat, acido means acid, phile means loving, means it requires both heat, high temperature and acidic conditions for their survival. And hyperthermophiles means more than extreme temperatures. Okay, and Pyrolobus fumeri, this holds the record for high temperature growth at 113 degrees centigrade. Okay, this is the famous example, Pyrolobus fumeri. It can grow at 113 degrees centigrade. Just remember this. Okay, these are halophiles, these are hot springs, and this is methanogens. Corarcheota, core means young. Okay, these are also called as Xenarcheota. This here the organisms were isolated from Yellowstone National Park, where pH is around 5.7 to 7 and high temperatures exist. Like Stomarcheota. These are chemolithotrophs. They utilize ammonia, or they oxidize ammonia, and they help in biogeochemical cycles. You have studied about carbon cycle, nitrogen cycle, sulfur cycle, etc., right? So these organisms, they are helping in running of these cycles. Without these organisms, you can't expect that cycles to be run in nature. Next, Nanarchaeota. This is old dwarf, meaning is old dwarf. Why we are saying it as dwarf means the cell size is very small. Okay, how much small? 1/100 size of E. coli. E. coli is a bacteria which is very small, and this nanoarchaeota is of 100th size of E. coli. So it is very, very small. It is about 0.4 micrometers in diameter and replicate only when attached to the surface of ignicoccus. Means replication. So only one organism is known from this phylum, that is Nanoarchaeum equitans. You can remember this name, Nanoarchaeum equitans. This slide is more important. So for what you have to remember about Archeabacteria is they are extremophiles. Basic thing is extremophiles, means they love extreme conditions. First important point. And then what kind of extreme conditions? It can be heat, it can be acidic conditions, or it can be alkaline conditions. And what are the uses? This thermostable DNA polymerase. Thermostable DNA polymerase. Can anyone tell me what is this? What is the function of DNA polymerase?
To read the DNA strand and synthesize the new
Yes.
strand.
Yes. What is the process? It helps replication.
Replication.
Yes. So thermostable DNA polymerases means the DNA polymerase, these enzymes which are stable at high temperatures. Right? Example, PFU DNA polymerase. It is used in PCR, and this enzyme is isolated from Pyrococcus furiosus. Produce enzymes like amylases, galactosidases, pullulonases that can function at 100 degrees centigrade. Means while cooking, baking, okay, we need this high temperature. Okay, so at that temperature also these microorganisms or these enzymes, they can function. Right? Apart from that, these microorganisms, they help in sewage treatment. They carry out anaerobic digestion. As we have seen, anaerobes are there in this group. They help in anaerobic digestion of this sludge, and they help in production of biogas and extraction of metals. acidophilic algae, Archeabacteria. They help in extraction of gold, copper, and cobalt from their ores, and they help in methane production. And as we have seen, they help in biogas production. Okay, methane they'll produce. So this was about another important point is they help in running of cycles, carbon, nitrogen, and sulfur cycles, etc. And as they are tolerating extreme conditions, researchers are more interested about these bacteria, and they want to see what conditions are helping them. And antibiotics, they host a new class of potentially useful antibiotics. Means we have a scope of getting new antibiotics from these archaebacteria. So this was about the archaebacteria. Now we'll take a 2 minutes break and we will discuss some important bits which were covered in previous question papers. So are you all ready to solve?
Yes, ma'am.
Meanwhile, if you have any doubts, you can ask.
Any doubt, my dear friends? Ashwini, Naresh ji, Sujata ji, Varsha ji, Sariya ji.
Yes, sir.
So Madhuri madam is asking about any queries regarding of today's class. So madam has clearly explained about the Archeabacteria, meaning of the Archeabacteria, and these characteristic features of the Archeabacteria, and what are the advantages of the Archeabacteria. So madam has clearly explained. Are you any queries? Okay. Madam?
Thanks.
Yes, sir. We are starting.
Okay. Okay.